with Moyna College, and thanks to both the principals for the MOU, more understanding. And today we have Dr. Mandal. He will talk about 1 hour for you students. So be attentive and interact with Madam whenever possible.
Okay, thank you, ma'am.
Without delay, yes, we want to start
Yes,
the session on time.
yes,
So,
yes, ma'am.
over to Dr. Acharji for introduction.
Okay, thank you, ma'am. So, we are very glad to have Dr. Sunandana Mandal. I would like to have the privilege to welcome ma'am and give a brief introduction. Dr. Sunandana Mandal is working as an assistant professor in the Department of Chemistry at Moyna College. Having a uniform first-class career, she completed her PhD degree from Vishwa Bharati in Soil Science and Agricultural Chemistry under the guidance of Professor Gautam Kumar Ghosh. In 2014, she participated in the summer study camp at Yunnan University in China as a Vishwa Bharati sponsored delegate. She has several publications to her credit, which are in very reputed journals at the national and international levels. She has also delivered lectures and presented papers in various seminars and conferences at the national and international level. She is also the reviewer of several international journals and a member of editorial team of ACTA Scientific Pharmacology, and also associated as a member of Indian Society of Soil Science New Delhi and Asian Council of Science Editors Giara, Dubai, UAE. So we are very happy to have you, ma'am. Ma'am will be speaking on the synthesis and characterization of novel dual capped ZNN nanofertilizers for facilitating nanoscale nutrient application in rice-based cropping system. So we are going to have a very interesting session. I would now like to request ma'am to please go on with the technical session. Ma'am, please.
Yes, thank you so much, madam, for this lovely introduction. A very good evening to everyone, all the faculty members of Department of Chemistry, Durgapur Government College, and other faculty members. Dear students, a very good evening. So, wait a second. So the topic of my today's presentation is the synthesis and characterization of novel dual-capped zinc nitrogen nanofertilizers for facilitating nanoscale nutrient application in rice-based cropping system. So why we have chosen this topic? Because zinc deficiency has been identified as one of the major health concern in today's scenario, and due to this effect children under the age 5 die annually because of this effect, and currently 2.7 billion population is affected by this zinc deficiency. This has been found to be acute for the rice consumers, that is the veg eaters, and zinc is found to be an essential constituent for a large number of enzymes, zinc finger protein, genetic materials, DNA, RNA. All these zinc plays an important role. So to overcome this problem, we will see the biofortification, which is considered as the most viable, maintainable, and inexpensive path among the other interventions to increase the grain zinc concentration. And biofortification can be of two types: agronomic biofortification and genetic biofortification. The first one is a less time consuming path and it is cost effective, and the later biofortification, that is the genetic biofortification, it takes more time and it is difficult to achieve. So we will go for the first one, that is the agronomic biofortification. It appears as a prompt solution to increase grain zinc concentration, and we can achieve this following two conventional methods. The first one is the soil application, and the another one is the foliar fertilization. So we see that when applied on soil, it undergoes various complexation reactions with soil, so less is available for the plant uptake. So this is the schematic diagram of by we can remove the zinc deficiency. We can go for zinc supplementation and food fortification, and there is biofortification as I already have mentioned. Two types are there: fertilizer application and genetic engineering. The fertilizer application involves these two methods. One is the foliar spray and another one is the soil broadcasting. So, in to address this problem, biofortification, specifically agronomic biofortification, we are considering nanoparticles as the fertilizer. So, why we have chosen nanoparticles? Because nanoparticles have high surface to volume ratio, hence they show distinct, unique chemical, biochemical as well as physical properties in comparison to their bulk counterparts. And these are 1 billionth of a meter, and it is expressed by the word nano. The aim of this present study is to cut down the use of chemical fertilizer, reduce the loss of nutrients, and maximize the crop yield. So here we have chosen nanofertilizers, and to increase the grain zinc concentration, we will go for the zinc nanofertilizers. So as we have earlier stated that zinc is an essential micronutrient for the human population. So we must go for the agronomic biofortification. But for the agronomic biofortification, zinc application has to be done. But in 25 years, it can happen that zinc may get depleted. So we have to use zinc fertilizers in a moderate way. Several bulk fertilizers are present. These are the zinc sulfate, zinc oxide powder, and some chelated zinc fertilizers, that is zinc EDTA, zinc liganosulfonate. But here we will consider for the zinc nanofertilizers for the effective nutrient delivery. In contrast to these bulk forms, nanotechnology has permitted the development of novel zinc type fertilizers because of their remarkable particle size and effectiveness and reactivities. So for the normal conventional fertilizer, we have seen two methods are there. One
Yeah,
is the soil
ma'am,
method.
sorry to interrupt you, but the
Yes.
slides are actually not moving. Can you
Oh,
please help us?
slides are not moving, but I'm okay.
Yeah, yeah, now it is moving.
Okay.
Yes, yes, ma'am. Yeah, students, can you able to see the slides?
Is
It's
it moving now?
yeah, Sneha.
হ্যাঁ ম্যাম।
Yes, ma'am.
হ্যাঁ এখন মুভ করছে তো?
হ্যাঁ ম্যাম, different methods of nano fertilizer applications যে স্লাইডটা আছে ওইটা এখন দেখতে পাচ্ছি।
হ্যাঁ
ওকে
হ্যাঁ
ওকে,
হ্যাঁ
মুভ করছে তো?
হ্যাঁ ম্যাম, এটাতে এখন স্টিক রয়েছে।
আমি কিন্তু মুভ করছি, দেখো তো, দেখতে পাচ্ছ?
না ম্যাম, এখন আর কোনো মুভ হয়নি।
আচ্ছা, significance of the present study?
না ম্যাম।
No, moving, it's not moving at all.
ওকে,
আচ্ছা, now it has come significant.
তো আমি এরকম যদি করি, আমি স্লাইড শো না করে যদি এমনি জাস্ট স্ক্রিন শেয়ার করি তাহলে হবে কি?
হ্যাঁ
তাহলে
হ্যাঁ
কি?
দেখা যাচ্ছে।
হ্যাঁ,
সব দ্য প্রেজেন্ট স্টাডি ওটা দেখা যাচ্ছে।
আচ্ছা আচ্ছা, হ্যাঁ, এটাতে জাস্ট আমি এমনি স্লাইড শোতে দিইনি, স্লাইড শো না করলে দেখছি মুভ হচ্ছে, তাহলে এটা করব কি?
হ্যাঁ হ্যাঁ
okay, select the slide only.
ওকে ওকে,
হ্যাঁ হ্যাঁ
ইজ ইট ভিজিবল?
হ্যাঁ এখন ম্যাম we
Okay.
are able to see the slide which is showing different methods of nanofertilizer application.
Okay, okay, thank you. So, as we had stated earlier, that in case of the conventional fertilizer, two methods were present: soil application as well as the foliar fertilization. But in case of the nanofertilizer application, different methods are to be employed because the conventional methods are no longer applicable because we face severe difficulties to for this. So in case of the nanofertilizer, if we consider surface broadcast, it will lead to drift losses. And another chance is that if we consider for the deep placement of the nanopowder, it can eliminate the drift hazard, but the nanoparticle may get adhered to the equipment surfaces, especially under wet conditions. So we can get the altered form. We will not get the fertilizer in the nano form intact. If consider the suspension of the nanoparticles in water used as soil drench, foliar spray, or nanoparticles dissolved into some ions, it can may degrade into its non-nanoscale structure. So soil we cannot use as the foliar spray. And if we consider it to be applied along with the macronutrient fertilizer, there remains size difference because the macronutrients are in the size range millimeter and the nano fertilizers are in the size range micrometer. So there is huge size difference. And apart from this, large scale field application of micronutrients that are required in small amounts results in non uniform distribution of the nutrients, which ultimately leads to unpredictable effects on the crop growth. So it is critical to develop strategies. So we have here employed a new strategy, that is to synthesize zinc oxide nanoparticles, which is binary capped with capping agents or stabilizers, and then we have coated these nanoparticles on the urea granules or NPK granules. So this way we can achieve our desired product and we can deliver nutrients in one go, and the macro as well as the micronutrients can be delivered together. So this is the new method we have employed. So significance of this present study. So this nanofertilizer which we aim to synthesize, it actually works efficiently to deliver nutrients at nanoscale, which will eliminate the environmental pollution, and the dual capping will control the particle size to obtain ultra small particles. And apart from this, the dual capping will enhances the stability of the synthesized nanoparticles. For the binary capping or the dual capping, we have chosen three stabilizers. These are NAC, that is N-acetyl cysteine, SAL, and urea. SAL, the sodium salicylate. So these three stabilizers we have chosen to cap the synthesized zinc oxide nanoparticles. By synthesizing this nanoparticle, we can achieve targeted nutrient delivery, and we can rule out the residual effects of the fertilizers to undergo reaction with soil components. So, zinc metal present in dual capped zinc urea nanofertilizers are less liable towards fixation by soil components, which will be an emerging novel approach by enhancing the zinc phytoavailability. And these capping agents, what they do, they change the chemistry of the surface layer of the nanoparticles, thus they stabilize them within the preparation medium. So, these zinc oxide nanoparticles, they remain intact. So this is why we have done the binary capping. Now, why we have chosen the zinc oxide as the zinc oxide nanoparticles? Because zinc oxide poses several benefits. These nanomaterials are rigid and hard, and hence they are a very good choice in ceramic engineering and manufacturing. These zinc oxide nanoparticles are widely used for medicinal purposes because of negligible level of toxicity and their biodegradability. These zinc oxide nanoparticles show promising usage as micronutrient fertilizers, and they can replace the conventional zinc salt fertilizers. And they possess higher physiochemical stability, high chemical coupling ability, and a wide range of radiation absorptivity. That is why we have chosen zinc oxide nanoparticles. These are the several applications of zinc oxide nanoparticles. As we see here, they can be used in drug delivery, they can be used in agriculture and farming, food processing and packaging, crop improvement, and they show antimicrobial activity. These are the three capping agents which we have used for the dual capping: urea, sodium salicylate, and N-acetyl cysteine. So what is the aim and objective of the present study? The aim of this present study is to develop novel nano-enabled fertilizer composed of urea and zinc, and the capping of zinc nanoparticles first with binary combinations of these three capping agents which I already have stated before being coated onto the urea granules. So after synthesizing, we will check the efficiency of these nanofertilizers in enhancing plant performance and nutrient delivery. in comparison with the standard bulk zinc fertilizers, which are known as zinc sulfate heptahydrate or nano zinc oxide. And we will also check the effect of biofortification of dual capped zinc nitrogen nanofertilizers coated urea through soil application at different growth stages on the rice crop, and several quality parameters of the rice grains such as starch amylose and crude protein content will be checked, and we also will find the zinc content of the rice grain. This is the synthesis technique, synthesis of zinc oxide nanoparticles at room temperature using wet chemical precipitation method. So in the right corner, we have given the amount of the capping agents required for the binary capping. So first we have taken 91 grams of zinc nitrate hexahydrate in 700 ml distilled water, and we have added two binary agents in the powdered form directly into the solution, and the solution was kept stirring, and then the pH was adjusted to 9 by adding sodium hydroxide, and the solution was kept stirring vigorously using mechanical stirrer. The final volume was adjusted to 1 liter, and the zinc concentration at this stage was 20,000 parts per millimeter. So we have synthesized dual capped zinc oxide, which will be purified by carrying out dialysis for 72 hours and changing the distilled water by every 8 hours. By freeze drying the entire dialyzed solution, the synthesized binary capped nano zinc oxide powder was obtained. Now the urea granules will be coated by these synthesized dual capped zinc nanoparticles using layering technique. So this is actually the preliminary idea. We haven't synthesized yet. This is the review work. We will synthesize it, and then comes to the characterization part. So the synthesized nanoparticles will be characterized using high resolution transmission electron microscope, and the morphology will be studied using scanning electron microscope. The zinc content in this dual capped zinc nitrogen powder will be determined using atomic absorption spectroscopy, and these are all the characterization. Now we will check the effectiveness of the synthesized nanoparticles dual capped by carrying out field trials. So for that, large number of soil samples will be collected from five selected blocks of Birbhum district, which are agronomically important, and 5 soil profiles will also be collected along with the surface soil, and these soils representing red lateritic soil of West Bengal, and the available nutrient status will be carried out. These selected soil profiles will then be analyzed for different zinc fractions present in soil, and we will find the correlation study will be carried out between these zinc fractions and different soil properties. So another characterization technique which we will employ is the is considering the zeta potential. It actually gives the surface charge values. So in case of the NaC containing nanoparticle, we can expect a negative zeta potential value because of the carboxylate ion which forms at pH 9. And in case of the ACl containing zinc oxide nanoparticles, which can we can get a positive charge because of the lower prevalence of negative charge in ACl, and this is suppressed by the higher prevalence of the positive charge of the urea. So this is the binding mode of the sodium salicylate. We can see there are 3 modes are present: mode 1, mode 2, and mode 3. So M1, M2, and M3 represents the metal atoms. So these 3 modes of capping of zinc oxide nanoparticles by salicylate. This is the capping of zinc oxide nanoparticles by N-acetyl cysteine. This is another capping agent. So this is why we have used capping agents. These are some benefits. It actually prevents agglomeration or aggregation. It controls particle size. It helps in the storage and stability. It provides functional group for attachment of biomolecules. This is also capping of zinc oxide nanoparticles. This is the schematic depiction of what we have, what our present study looks like. So first we have synthesized zinc oxide nanoparticles, which is dually kept, and then these nanoparticles were coated onto the surface of the urea granules, and then we will go for the characterization. We already have discussed some characterization techniques, and then we will find its effectiveness in rice crop, whether it is helpful for increasing zinc content or not. These are the field experiments. Three locations for the field experiments of biofortification of zinc in rice grain was selected based on the deficient zinc, marginal zinc, and sufficient zinc range. Field experiments during 2 seasons, wet and dry, will be carried out, and we will determine zinc concentration in the grain. The different fractions like brain, embryo, and endosperm, and as well as the whole grain in 3 locations in the district. Soil applications of dual kept zinc nitrogen nanofertilizers will be conducted at various growth stages. These growth stages are stem elongation, boot, milk, and dose stages. To evaluate the outcome of the timing of the soil zinc application, yield will be recorded. Quality parameters like starch, amylose, and crude protein will also be analyzed, and the localization of zinc within the rice grain will also be carried out using laser ablation technique. This is the research design and methodology. This is the details of the field experiment. So the crop is rice. 19 treatments are there. 3 replications, and total number of plots is 57. These are the different treatment details, the 19 treatment details, depending on the time of application of this nanofertilizer and the percentage of urea. This is the plan of work, schematic and graphical representation of the plan of work. So what we can expect out of this study? So this research is primarily aimed to obtain relevant scientific information of zinc status in the red acidic soil of West Bengal, and the biofortification of dual capped zinc nitrogen nanofertilizers applied in soil in polished as well as brown rice. Now, rice is considered as a poor source when it comes to the bioavailability of zinc, and the biofortification of rice through dual kept zinc nitrogen fertilization represents a promising solution to overcome this problem, and scientific reports will be generated out of this study. So we haven't synthesized the nanoparticle yet, but we have collected soil samples, and we have analyzed different macro and micronutrient status, and the correlation study has also been carried out. So this is the chemical analysis of the experimental soil. This is the map showing the location of the study area. These are the different soil parameters analyzed: soil pH, electrical conductivity, particle size distribution, soil organic carbon content, different nutrients, different macronutrients, namely nitrogen, phosphorus, sulfur, and different micronutrients, namely zinc, iron, manganese, copper, and the different zinc fractions in soil, that is water soluble and exchangeable zinc, organically complex zinc, amorphous sesquioxide bound zinc, crystalline sesquioxide bound zinc, manganese oxide bound zinc, and the total zinc present in soil solution. These protocols will be followed, which is given here. And these are the sequential fractionation procedure which have been employed for determining different zinc fractions present in soil, given by Murthy and modified by Mondal. These are the different plant parameters which we will analyze: chlorophyll content, nitrogen, phosphorus, potassium, and zinc content, and some quality parameters will also be studied. The statistical analysis will be carried out using analysis of variance technique, and the correlation analysis will be carried out using Pearson's method, and at the level of 1% and 5% level of significance. These are the classification of soil pH and soil organic carbon content values. These are the classification of the total soluble salts. These are the rating chart for macronutrient and micronutrients in soil required for plant growth. These are the soil profiles collected at three different depths of the agronomically important areas of Birbhum district: 0 to 15 centimeter, 15 to 30 centimeter, and 30 to 60 centimeter. These are the five soil profiles. The latitude and longitude is also given here. These are the physicochemical properties and available macronutrient status of the collected soil samples from selected blocks of Birbhum district. We find the textural class as the silt loam, silt loam, and respectively. Now we come to the correlation analysis. So we see that pH correlates significantly and negatively with available nitrogen, potassium and sulfur, whereas significantly and positively with available phosphorus. This actually implies that at higher pH these nutrients, namely nitrogen, potassium and sulfur, are less available, whereas more plant available phosphorus is present at higher pH. So inverse relationship is present between available nitrogen, potassium and sulfur with soil pH. On the other hand, significant and positive correlation is present between soil organic carbon content and different macronutrients, which actually indicates that organic matter provides ligands which form soluble complexes, and these soluble complexes are readily available for the plant uptake. Now here the table 12 represent the regression coefficients of significant correlations of soil available macronutrients with different soil physicochemical properties. R square value gives the interaction between these parameters, which are denoted in the left side of the table. These are the scatter plot. Scatter plot is the indicative of the interaction between the macronutrients and the soil physicochemical properties. So as we see here that a negative correlation is present between available nitrogen and pH. So similarly from this graph we can also explain this relationship, the negative association between soil pH and available nitrogen. Similarly, positive association present between available nitrogen and soil organic carbon content is presented in figure 3, which is the scatter plot showing the relationship between available nitrogen and organic carbon content. Similarly, we can explain similar relationship with a scatter plot, what we have already discussed in this correlation analysis. So we come to the results of the macronutrient analysis. So the red acidic soils of Birbhum districts are characterized under strong to neutral in soil reaction, non-saline in nature, and silt loam to silt in soil's texture. The soil samples are having low to medium soil organic carbon content. As we have shown earlier that soil organic carbon content, that is SOC, significantly and positively correlated with all macronutrients, which suggests the need to manage optimum amounts of soil organic carbon content in order to maintain adequate supplies of essential nutrients for the plant uptake. Now we come to the available micronutrient status. So the table shows the different micronutrient status. These are the correlation analysis. So in case of the micronutrients, we also see significant and negative correlation between the soil pH and different micronutrients. So inverse relationship is present, whereas significant and positive relationship present between organic carbon content and different micronutrients, which actually indicates that soil organic matter provides soluble chelating ligands which form soluble complexes or labile complexes with these micronutrients, which are readily available in soil solution. Hence plants can take up these essential nutrients easily, and they do not form any complexes which are non-labile in nature. These are the multiple regression equation indicating the indicating the importance of the soil parameter. This is also the scatter plot which we have mentioned earlier in case of the macronutrient status, showing the association between different soil physicochemical properties and the available micronutrients. So, from the surface soil sample analysis, we can say that 76% of the collected soil samples were found a deficit in available zinc, 20% were having marginal, and 4% were found sufficient in available zinc. And the results of the stepwise multiple regression analysis showed that the major contributions towards available micronutrients are coming from soil pH and soil organic carbon content. So now we come to the soil profiles. We already have mentioned that we have collected 5 soil profiles at 3 different depths, 0 to 15 centimeter, 15 to 30 centimeter, and 30 to 60 centimeter. So we see that the higher values of sand at the surface layer and lower value with increasing trend down the depth. But in case of the silt and clay, lower values are present in the lower level, whereas leaching occurs and we get higher percentage of silt and clay at lower depths. And the textural class has also been mentioned here, which is silt loam, silt, silt, silt loam, and silt for these 5 soil profiles respectively. These are the chemical characteristics of the soil profiles. As we see that in case of the pH value, higher values are obtained at lower depths, whereas lower pH values were recorded in surface layer. But the reverse trend has been followed in case of the organic carbon content and available macro and micronutrients. That is, higher values were recorded in the surface layer and lower value at lower layer, which might be due to the fact that higher organic matter present in the surface layer, which provides soluble ligands and lesser movement down the profile, and hence lesser availability of these labile ligands at lower depths. These are the micronutrient values which I already have talked earlier. So this is the correlation we come in case of different micro and macronutrients. We see significant and negative correlation with soil pH values, significant and positive correlation with the organic carbon content values. So there is inverse relationship present between pH and these available nutrients, whereas a significant positive correlation is present between soil organic carbon content and these different nutrients. So we see that these available macronutrients, that is N, P, S and K in general, decreased with depth in all soil profiles. Higher values of macronutrients in the surface layer were observed in the profiles due to high organic carbon content, which was mostly influenced by plant residues on the soil surface and less movement down the profile. Similar pattern is observed in case of the micronutrients. So, by supplementing deficient nutrient by applying chemical fertilizer or organic manure, it can improve the soil health and promote sustainable paddy production in these soils. And these informations generated can be helpful for both the deep and shallow rooted crops, and these informations will be helpful for our present study. We will choose 3 locations depending on the zinc status value, that is the marginal range, sufficient and deficient range, and then we will apply our zinc oxide nanofertilizer, and we will carry out the field experiments. Now these are the different zinc fractions present in soil. The first one is the water soluble and exchangeable zinc, then comes the organically complexed zinc, then comes amorphous sesquioxide bound zinc, then comes crystalline sesquioxide bound zinc, manganese sesquioxide bound zinc, and residual zinc, and the total zinc represents the total zinc present in soil. So the first two fractions, that is water soluble and exchangeable zinc as well as organically complexed zinc, these two fractions are the most labile, and these when zinc present in these two fractions, they can readily be taken up by the plant because they forms soluble complexes with ligands or chelates. But in case of the amorphous sesquioxide bound zinc and crystalline sesquioxide bound zinc, zinc is not that much present for the plant uptake because they are non-labile in nature. And in case of the manganese oxide bound zinc and residual zinc, zinc present in this fraction are not labile in nature. These actually these two forms represent the non-labile pool. So these are the intercorrelation between different forms of zinc in soil solution. So we see that there is significant positive correlation present between different zinc fractions in soil, which actually represents that there remains some dynamic equilibrium between these different zinc fractions. So somewhat zinc is transformable into one another form, but it requires time or under vigorous reaction condition. These are the multiple regression equation. So we see that the pH and organic carbon content are the major two contributors, which actually makes the zinc more available for the plant uptake. So results showed widespread zinc deficiency of available zinc along the soil profiles, which might be due to the presence of higher concentration of zinc in stable fractions and slow release into the soil solution as available form. And the correlation and regression analysis showed that pH and organic carbon are the soil properties which are related to the distribution of zinc in selected soil profiles. So this is the experiment 2, that is the biofortification of zinc through sources and levels of zinc to kharif and boro rice. This experiment has not been conducted yet. This is a very basic work we have carried out until now. So this work is funded by Science and Technology and Biotechnology Department, Government of West Bengal. These are some of the references I have used. These are the publications, some of my publications. So I'm very much thankful to Dr. Debnath Palit, Principal, Durgapur Government College, Dr. Snigdha Chandra Madam, Head Department of Chemistry, Durgapur Government College, and all other faculty members of Department of Chemistry, Nivedita Acharya Madam for her lovely introduction, Dr. Shapan Kumar Ghosh, Head Department of Physics, Durgapur Government College, and all other faculty members. I am also thankful to Dr. Dilip Kumar Panda sir, Principal, Moyna College, and all other faculty members. I am thankful to Department of Science and Technology, Biotechnology, Government of West Bengal for their financial support. I am thankful to Professor Gautam Kumar Ghosh, Department of Soil Science and Agricultural Chemistry, Vishwa Bharati. So thank you. You can ask me if you have any question.
Yeah, so many thanks to you, Sunandana madam, for such a nice lecture, a comprehensive one, and introducing the soil research to our students. So students can ask any questions or queries if they have, or any faculty member or students can ask. You can also write in the chat box, or anything, any views also you can give.
I have one question, Sunandana.
Yes, please.
You mentioned the pH of Rampurat is basic at the level of 30 to 60 centimeter. At
Okay, just let me open this slide.
7.07. Yeah.
Yes. Hmm.
Rampurat.
Yes. Yes, this is the slide. Yes, madam, please.
Am I right? Rampurat
Yeah, it's
showing pH.
Yeah, it's neutral to acidic in soil reaction. The pH values we have collected, it's neutral to high acidic in soil reaction.
Where you mentioned it 7.07 like that.
7.07. Yeah, it's 7.37. It's the range.
7.37. The interval of sampling, may I know?
Interval of sampling. No, this is for, only one time sampling we have done. Surface soil samples, one time collection, and then the soil profiles. It was also one time collection.
No, after application of fertilizer.
No, we haven't applied it. We only have collected the soil samples, the surface soil sample as well as the profile soil sample to get an idea of the zinc status of this block. And then we will carry out the research. We will apply it on soil and then we will see how the results resonate with the fertilizer application.
Okay, okay. Very much interested in seeing the dynamics of the
Yeah, because
nanofertilizer in soil.
that work has not yet been done. So I will do it and hopefully I can share it with you in future.
Thank you.
Welcome, madam.
Yeah, thank you, ma'am, for the nice question. Any students can ask, or we also have a feedback link. We will be sharing it with our students and faculty members. And ma'am is with always with us. We will be communicating all of the questions to her. So, if there are not any questions, so we can just wind up the session by thanking Sunandana madam for such a nice faculty exchange lecture. Actually, teaching is actually exchange of ideas, and between two colleges, this is just the beginning. We will be organizing many more events, and we will be helpful. It will be very helpful if we can get your help and assistance for our students, madam. So,
Thank you.
yeah, so thanks to all for joining such an interesting lecture. So we can just wind up the session. So good night to all.
Thanks everyone. Yeah.
Good night, everyone.
In our yeah, good night. One more thing,
Yeah.
Sunandana.
Yes, ma'am.
Our
Yes, ma'am.
four-year research program is will just start from, I think, this session or the next session.
Okay.
So honors, no, some kind of research program will be there for UT
Yes,
students.
yes, yes.
There also we can have some project, small work on agricultural chemistry.
Yeah,
We
yeah,
can
yeah,
your
sure.
help there.
Yeah, definitely. Definitely.
Okay,
Please let me know, madam. I will be happy to help.
okay, good.
Yeah, many
Thank
thanks
you
to
so
you,
much.
ma'am.
Yeah,
Yes,
Welcome, madam.
for extending your help for our students. So thanks to all. We will be joining in another interesting lecture.